Hi, everyone. It's a great pleasure to introduce myself and our research program at this conference. I'm currently the Marian and Harry Caper Chair Professor in Cancer Research at Norris Comprehensive Cancer Center of USC. I was trained at the Rockefeller University from 2002 to 2006 before joining the University of Michigan, Ann Arbor as a faculty. 2 years ago, my lab relocated to USC Keck School of Medicine. In my lab, we study epigenetic regulation in development and diseases. Epigenetic is a main contributor to cell identity. For example, all the cells in our body share exactly the same genetic information we inherited from our parents. But the skin cells are very different from heart cells, and brain cells are very different from fat cells. Our research is really to understand the epigenetic basis of these differences at the molecular level and how its dysregulation contribute to human diseases. Specifically, we study the post-translational chromatin modifications and their function in regulating gene expression, cellular metabolism, and 3D chromatin structures, and to translate our basic science to potential therapeutics. Our research is well funded by several NIH grants, as well as by funding from private foundations. One of the projects in our lab is to focus on histone methyltransferase, MLL, which methylate histone and regulate transcription activation. ML was initially identified as to be translocated in a subtype of leukemia to generate ML fusion proteins. It has been established that both wild-type ML and ML fusion proteins contribute to leukemogenesis and therefore serve as potential therapeutic targets. In addition to ML-rearranged leukemia, ML amplification and overexpression have also been reported in a broad spectrum of cancer. We have first reconstituted the minimal ML complex required for its methyltransferase activity. Later, we carry out extensive biochemical and structural studies to understand how ML activity is regulated on chromatin, which is its physiological substrate, and how this activity can be exploited to develop ML inhibitors. This study have established several defining features of the ML complex that distinguish it from other chromatin-modifying enzymes. Based on the basic biochemical studies, we have identified novel strategies to inhibit ML activity. We have developed both small molecule inhibitors and monobodies to target ML interaction with a protein called WDR5. This inhibition effectively disrupts ML complex integrity and blocks leukemia cell growth both in vivo and in vitro. It represents one of the several mechanism-based strategies that are being actively pursued for drug development. We have ongoing effort to develop other ML inhibitors for cancer treatment. We are looking for both industrial and academic collaborators for preclinical and clinical development of ML inhibitors. We are also open to partners to identify and validate other potential epigenetic targets for cancer treatment. Thank you.